This protocol allows the derivation of sympathetic neurons from human pluripotent stem cells. These cells will enable scientists to study human disorders that affect the sympathetic nervous system.
This technique allows the derivation of sympathetic neurons in defined and feeder-free medium conditions at a high scalable efficiency. It results in electrically active neurons in 20 days. This technique can be applied to diseases caused by a dysfunctional sympathetic nervous system. It also can be used for disease modeling, regenerative medicine, and drug screening.
These cells could be used in vitro to innervate any tissue within a co-culture system. For example, for the study of cardiac tissue regulation.
When the human pluripotent stem cell culture reaches 80 to 90% confluency, big colonies with smooth, bright edges should be observed. For splitting, wash the culture with PBS before treating the cells with 4 milliliters of 0.25 molar EDTA at 37 degrees Celsius and 5% carbon dioxide. After 2 minutes, flush the plate with 10 milliliters of E8 medium and transfer the detached cell suspension to a 15 milliliter conical tube. Then transfer 500 microliters of cells into a 9.5 milliliter aliquot of fresh E8 medium and plate the stem cells onto new vitronectin-coated 100 millimeter dishes. One day before differentiation induction, coat the appropriate number of 6-well plates with 2 milliliters of